Hello everyone. Welcome to the practical sessions on bioinformatics. In this session, we'll be discussing data retrieval from protein data bank database. As you know, protein data bank contains the 3D structures of proteins, and that PDB files or protein data bank files can be retrieved for visualization in your system using using the methods described in this practical. Now, in the list of the practicals, you can see the data retrieval from PDB link. You can open that one. So once it is open, see the aim of the or objective of the practical to retrieve the structure of the proteins from the protein structural database PDB, for which you require to open the RCSB website. So this is the rcsb.org website. Here there will be a search box. You can type in the name of the protein of interest. For example, insulin, and search. So it will give you the list of the entries present in the database. And you can also filter the results within the source organs, so for example, Homo sapiens or Bos taurus or any organism, or for example, Escherichia coli. So insulin expressed in E. coli will be there, for example, 14. So if you are looking for some insulin that is expressed in E. coli, you can click on this one. So it will be filtered, and based on the different experimental methods also, there are structures determined using X-ray diffraction or NMR, electron microscopy. So various experimental methods were also described here, and you can filter the results using such experimental methods also. Okay, now here we have various results that is shown. So the 40 structures from E. coli. So it is filtered by clicking on this link. And if you are interested in a crystal structure of a human insulin degrading enzyme, so note that it is not insulin, it is an insulin degrading enzyme, so which is not our interest. So these are not exactly the same. So in that case, you can go back, clear all filters. Let's see Homo sapiens. Say insulin-like growth factor is not exactly that we are looking for. We are looking for insulin sequence. Insulin-like, insulin, structure of insulin presence of urea at pH. So this is an insulin. Maybe you are interested in this one. So look at the descriptions and see that it is insulin itself, not insulin inhibitor or insulin binding protein or so some other proteins related with the insulin, or some other structures with the term insulin in the structure will also be in the title will also be listed along with that one. So you have to select insulin itself. So once it is open, you can see that the linear visualization of a 3D view of that structure is actually shown on the left side panel. This is the ID, PDB ID. which can be used for downloading the sequence using various softwares. So this is the DOI number of the entry, description and title. Classification is a hormone from Homo sapiens. Release date and this is a structure determined by X-ray crystallography, X-ray diffraction, and these are the parameters. Then this is a paper published or scientific literature in which this structure is published. Then various other information including the macromolecules available, for example insulin A chain, various chains, and total length of the sequence is 21. This is the B chain, total length of the sequence is 30. These are the small ligand molecules interacting with that chain. Experimental data, validation details, entry history. So a lot of information, annotated information is available related to the structure. So once you go through all these details, you can download the files. You can display the file, or there will be a, for example, if you want to display this in a 3D view, you can have a Java supported browser which will open the results. But anyway, it is not required right now. You are trying to download this structure, for which you require to download the file, and here there will be various options. If you are looking for FASTA sequence of the protein, you can select this one. But if you are looking for a PDB format, you can click on PDB format. So the file in PDB format will be saved in your system. So this is the file that is downloaded. If you have a structure visualization software, then you can open it in that software. In my computer, Maestro or professional softwares like Schrodinger is there, so which will open that file, or you can use such softwares like Rasmol or PyMol for opening this file, and that will be discussed in another practical. So now you downloaded the structure, and finally you can visualize it in Rasmol. Let me see whether Rasmol is installed. So if Rasmol software is not installed, then you have to download the Rasmol software and visualize it, and that will be discussed later in another practical. So in the practical, you have to finish the download. Then after installing the Rasmol, you can visualize the structure in Rasmol software. Okay. So you can see that in PDB download, the previous part we discussed is the structural summary, and here we have 3D view option here. And if you're taking that option, you can see the 3D visualization of that insulin gene, or sorry, insulin structure in the visualization software in the web interface itself. Or you can download the Rasmol, but even without downloading the Rasmol, you can visualize the 3D structure and rotate the structure in the web interface. So this is the annotations, family annotation, domain annotation, and other related function predictions, etc. Everything available in the website. Then these are the experimental details, the X-ray crystallography parameters and diffraction details of the structure prediction. This is the sequence information, binding size and various and sequence information is there, and even you can download the sequence from here. And this is the entire genome sequence information from Homo sapiens, from the start and end points of the gene in the human genome. It is also marked in the human genome in a graphical window. So all the annotations, experimental details, sequences, and position in the genome, etc., were available in the PDB database. Also, you can download the files. So you can go through all these tabs on the top side for getting the structural summary, 3D view, annotations, experimental details, sequences, and the location in the genome. Okay, thank you.